very much for joining us today, John, Helen, Oana. I'm here with my fellow committee members, Søren Friis, Director of Global Customer Relations, and Tim Strasmeyer, Director of Scientific Operations at Nanny & Inc. We were really impressed with your proposal, and before we get started delving into a bit more about your project and about the Medicines Discovery Institute at Cardiff, to make this all a little bit more relaxed, we have some great news for you. John, Helen, Emyr, Oana, you are the winners of the 2023 SyncroPatch 384 instrument grant. So congratulations.
Congratulations.
Wow, that's fantastic.
We would love to hear a bit more about your project and a bit more about the Medicines Discovery Institute, if you've got a few slides where you can explain this.
Yeah, so the Medicines Discovery Institute is this gaggle of people here. We're a multidisciplinary team of 40 biologists and chemists combined, and support staff. So our mission is to develop novel medicines for unmet need. We've had a focus initially on mental health. We're now moving into degeneration, focusing on Helen and Emyr's targets. And we also have a little bit of effort on cancer. And we'll cover these things very briefly in later slides. We go all the way from target identification, target validation. So this is work that we'll do along with academic colleagues. So Helen, Emyr, other people in the Neuroscience and Mental Health Innovation Institute do a lot of the genetics of schizophrenia, for example. And so we can work with them on target identification, target validation. And then we go all the way through to our flagship project, which is an AMPA receptor modulator project, which is in phase 1 and going clinical trials as we speak. This is funded by the Wellcome Trust. And this is a project that we've driven all the way through from lead optimization all the way into the clinic. And again, we'll touch on that briefly a bit later. There's a fully integrated drug discovery team. We have infrastructure to support, like I say, activities from hit identification all the way through to early clinical studies. Now, we don't have all that capability within the Medicines Discovery Institute, but we are for our drug discovery grants, we do collaborations externally. So people like Jerry Lambert in University of Dundee, Slight Electrophysiology. And then we'll do things like Uli Zeilhofer for our pain target in Zurich. Clinicians throughout the world that are associated with these various projects who have clinical input as well. Our physical labs comprise state-of-the-art chemistry labs. So these are 16 fume hoods. So that's nominally 16 chemists working a variety of targets. And we have our biology labs. Our biology labs within there, MDI, which are also complemented by Emyr's labs doing the lysosomal target stuff. And again, more about that later. So within our labs, we have assay development activities. We have X-ray crystallography for structurally enabled targets. We have medicinal chemistry, in vitro DMPK, so this is macrosomal turnover, things like that. Things like in vivo pharmacokinetics will contract out, and in vivo behavioral assays on a case-by-case basis, so for schizophrenia targets or whatever, we can contract those out. EEG activities will contract out. So we have a core of stuff that goes on in MDI, but we contract out key bits of work to contract research organisations. And a key partner in a lot of our ion channel targets is SB Drug Discovery up in Glasgow. So this is who we are, how we're organised. So myself as a director and Simon Ward, who looks after medicinal chemistry. So we're the sort of drug discovery people, complemented by Richard Angel and Michael, who are medicinal chemists, but also have other responsibilities within the group. Oana, who's not shown on this slide, is responsible for all the ion channel biology stuff on our GABA projects, for example. We have Jenny, who looks after project management, and Emily, who's recently left us, but we have an integrated project management scheme in place, and that's complemented by the academics at Cardiff University, and most notably on the call today, Helen and Emyr, who are the world-leading experts on lysosomal biology. Ben Bax, who is a structural biologist. Helen Jones is a medicinal chemist within Cardiff University. Georgina and Tom have an interest in DNA damage repair mechanisms. So again, we have a complement of drug discovery expertise, so myself and Simon, and target expertise tapped into with Helen and Emyr for lysosomal targets and other therapeutic areas as we consolidate the institute. So people like Tom and Georgie, DNA repair. We've also got interest in cancer, which we're growing again with academics within Cardiff University. So we are the drug discovery institute, really, that complements and helps translate the academics' basic knowledge into patient impact. Next slide, please. Please stop me at any point if any of this needs further elaboration. So our model is basically to identify an unmet need along with the academic scientists at Cardiff. So our initial focus has been mental health. That's complemented very much by the Neuroscience and Mental Health Innovation Institute at Cardiff University, which is a major strategic investment along with the Medicines Discovery Institute. And those neuroscience targets have now been expanded into neurodegeneration along with Helen and Emyr, and also the Dementia Research Institute at Cardiff University. The way we work is we use what we call our engine rooms. So these are a group of scientists that are biologists and chemists that are funded by Cardiff University. And this is the critical difference between what we do and what a lot of other academic groups do. We have Cardiff University funding, which funds people and funds resources, so can do contract research, organisation work. And that really is the engine room of the whole institute, which creates data packages, which will then go externally to the MRC, Wellcome Trust, commercial partners or whoever to try and get funded. So this group does the hit finding to find the chemical starting points, and we can also pick up additional chemistry starting points that have been left behind in the literature. I think one of those, Ali, you might be familiar with the DS2 delta selective GABA receptor modulator, which came out of Turnings Park. It was published, and then we wrapped that up into a story, did a bit of early SAR around that and got that funded. We did a similar thing with our glycine project, picked up a published lead from New Centis and did some early exploratory SAR, again with this engine room resource. And that engine room resource also tapped into the SB drug discovery capability for the ion channel work. And I think a key thing to emphasize is that we do have resources to dedicate to support SyncroPatch. So it's not as though we would have a piece of kit and then it just stand there collecting dust. We do have pairs of hands that we can prioritize to devote to this.
Yeah, this is definitely important. It's important to have those resources available and sort of allocated so that you can get the most out of it.
Yeah, and these are flexible resources. We allocate them as we see fit. This is the directors and me, Simon, Helen, Emyr, Oana would discuss where the next focus of the engine room needs to be, and we already have a really good idea. Well, we know already there's an ion channel, a lysosomal ion channel we want to focus on, and we're putting resources onto that. So we do have that flexibility to devote resource, both hand power, manpower and cash to particular projects, and then submit a project proposal to MRC, DPFS, or Wellcome Trust, or whoever. If we've got a data package that looks interesting, Helen and Emyr are really very well connected into lysosomal storage disorders and lysosomal patient organisations, so that's a great resource as well. And the ultimate aim of the Medicine Discovery Institute is to commercialise these projects. So we realise that we'll never have the funding to get these things onto the market to make a patient impact, but we'll generate a high quality data package, intellectual property, so the patents that will support commercialisation, either into a project into a spin-out company, or just our license the project to a commercial partner that will then take it on into the clinic. Next slide, please, Helen. And this is just a summary of our portfolio that we have at the moment. So the flagship thing, the AMPA project at the top is in phase 1. A whole gaggle of GABA modulator projects, which Oana has been driving thereafter. We've got the acid ceramidase shown there as a lysosomal target, but obviously, as you'll hear from Helen, there's a huge potential for ion channel modulators of lysosomal function, which is our next MDI strategic focus, which really complements this SyncroPatch scheme. There's a couple of blue things at the bottom for just to show that we do dabble in cancer. So we've got a project funded by the MRC that starts in January for acute myeloid leukemia. We've got a Wellcome Trust funded cancer project that was commercialized, so we licensed that to a South Korean company. And of note at the bottom, respiratory syncytial virus. That was our very first project that we worked on when we first started the group at the University of Sussex. That was probably 2014, so about 8-9 years ago. We work on a Wellcome Trust funded project, identified a molecule that was then taken on by a partner company, Reviral, and that was sold last year to Pfizer for $525 million. And the point there being that we can walk the walk as well as talk the talk. We can produce high quality molecules that get the way into the clinic and have legs on them. So the SyncroPatch utility within MDI very obviously will support our ongoing ion channel projects. So those are currently driven by Flipper. And the reason why they're driven by Flipper is because the cycle time and cost of doing electrophysiology SBDD means that we can't use ePhys to drive the project. Those ePhys assays have to be more icing on the cake experiments. But we think if we had an in-house SyncroPatch, we could actually drive the medicinal chemistry, which is obviously hugely more powerful. And the next use of a SyncroPatch would be target validation, hit finding for novel lysosomal targets. So this would enable our ability to build up this whole lysosomal biology area, which is driven by Helen and Emyr, which has huge potential.
Okay, so I'll take over at this point. And so we're planning to mostly use your SyncroPatch, as John said, to essentially discover new tools to enable research into lysosomal ion channels. So if you like, the lysosome has numerous ion channels and ion transporters. And so it's kind of like the final frontier of the ion channel world, because most of them are very poorly characterized. And they're poorly characterized because lysosomes are difficult to work with. Obviously, they're intracellular, they're really small, they're acidic, they have like a calyx around the membrane, which can make getting tool molecules to the ion channels difficult. But there has been lately a lot more interest in lysosomal ion channels. So they've been shown to be important in several common diseases of aging, which I'll get onto in the next slide. But this research is being stymied essentially because there's no good way. For most of them, there's no good ways to selectively modulate channel activity with small molecules. So then the transporters and channels you can see in this diagram here are not, there's not a comprehensive set of lysosomal channels and transporters. There are more of them than this, but very few of them have selective and potent agonists or antagonists. So you're like the only channels which have selective agonists in the lysosome are TRPML1, TPC2, and the BK channel. But of course, the BK channel is also found on the plasma membrane, and the same small molecule will work on both. So in terms of actually activating just a lysosomal channel, you're down to just TRPML1 or TPC2. The situation not a lot better with antagonists. So again, TRPML1, TPC2, and you can also add TPC1 and the vacuolar ATPase into the mix. So our plan is to use the expertise and the resources that we have here on ion channels, on lysosomes, on electrophysiology, and the resources that we have within the NDI to discover new tools to enable research on these channels. So we can work out exactly what it is they're doing in disease and whether they can be drug targets to help with neurodegenerative disease. So this is just an indication of the extent to which lysosomal ion channels are involved in neurodegenerative disease. So in the several GWAS that have been carried out lately for both Alzheimer's disease and Parkinson's disease, these have both found large numbers of lysosomal genes. So it's indisputable that lysosomes are important in the pathology of both Alzheimer's disease and Parkinson's disease. But if we look specifically at lysosomal ion channels and transporters, there are two identified risk genes for Alzheimer's disease, TPC1 and SLC24A4. But the role which these proteins play in Alzheimer's disease is not known, and there are no good tools really to try and find out what these are. We have a long-standing interest in the role of lysosomal acidification in diseases of aging. And it's been shown in familial Alzheimer's disease that reduced function of the vacuolar ATPase leads to lysosomal deacidification, and that this deacidification contributes to disease pathology. So that opens up the possibility that both the VATPase and also other ion channels which regulate lysosomal acidity, such as TMEM175 or CLCN7, could also be potential drug targets for Alzheimer's. But for TMEM175, CLCN7, and for activating the ATPase, we have no small molecule starting points. So lysosomal involvement is possibly even more obvious in Parkinson's disease, with the GBA1 connection, that being a lysosomal enzyme. But there are lysosomal ion channels involved as well. So the lysosomal channel ATP13A2 is a rare genetic cause of Parkinson's disease. So mutations in this channel cause a familial version of Parkinson's disease. Whereas, as I'm sure you know, TMEM175, which is a lysosomal ion channel / transporter, has been identified as a PD risk gene. And I'm very aware that you guys have found modulators of TMEM175, but I hope that you won't be offended when I tell you that these are not particularly selective for TMEM175. So moving on to things which are a bit a bit less mainstream, we also have an interest in the CLN3 channel, which Emyr identified as a lysosomal potassium channel. So mutations in this channel cause CLN3 disease, which is a neurodegenerative lysosomal storage disorder, but it's also been identified as a copy number variant in schizophrenia GWAS. So there's a link between CLN3 disease and a more common mental health issue, which may increase its attractiveness, if you like, as a good target for getting commercial investment. And lest we forget where Emyr and I started off, we're still very much interested in developing therapies for the lysosomal storage disorders, which are caused by mutations in these channels, because this is a huge area of unmet clinical need. Most of the lysosomal storage disorders have no available therapies whatsoever, and we're very keen to see if we can do something about that. And so finally, just like to give you a brief run through of essentially what our plan would be for each target, which is we'd first create the cell lines, which we have done for several channels, and then establish the conditions necessary for doing automated EFIS on the plasma membrane in these channels. And because Cardiff University also very kindly bought us a PortaPatch, we may use the PortaPatch because of its, essentially it's a lot cheaper and gives us greater flexibility to first determine the assay conditions and then attempt to transfer those over to the SyncroPatch, so we can have assays using any available tool compounds that might exist for the channel already to show that we can both activate and inhibit these channels. Once we've got the assay transferred over to the SyncroPatch, then our plan would be to screen a focused ion channel library, with a decision of the library to be made around the known characteristics of the channel, to see if we can identify novel molecules. Once we've identified them at single concentration, we determine a concentration-dependent effect by doing concentration response curves, and that of the best compounds from concentration response curves, we would utilize the medicinal chemistry resource within the NDI to identify commercially available related molecules, so that we can form structural activity relationships by buying molecules, so do SAR by catalogue, to see if by doing that we can find a better hit than what we originally found in the library. At that point, we would move on to our confirmatory assays, just to make sure that the best molecules that we found in the SyncroPatch screening are doing what we thought they should be doing in cells. So that will give us a nice data package, which we can then use to apply for external funding to develop either better tool molecules or ideally, because novel therapies for the mental health or neurodegenerative disorders that we think that these channels would be beneficial for.
Okay, wonderful. Thank you, Helen, Emyr, John, Oana. That was a really nice presentation. And yeah, like I said before, we were really impressed with the kind of ideas that you have and the lysosomal targets. So, I mean, one question I had, I mean, you plan to use the SyncroPatch really for overexpressing cell lines at the plasma membrane. Would you have any plan to also try the lysosomes on the SyncroPatch, or just on the surfer, or I guess PortaPatch maybe?
Maybe. Yeah, and that's a good question. So far as screening goes, I think we would stick with plasma membrane, just because, if nothing else, the feasibility of generating the number of swollen lysosomes that would be needed for assay is going to be quite difficult. Mm-hmm.
Yeah, what's the minimum size that you could patch?
Well, we have got these special plates coming out, which have a really small hole, and we've been doing also some preliminary experiments with lysosomes. So we know we can patch lysosomes. Would you know the smallest size, Tim or Søren?
Mm-hmm. Yeah, the smallest hole size for now is around 1 micrometer. in diameter.
That's
And
right.
that-
I think we have groups that are working with the 1 micron and the 1.5 micron, trying to get a lyso-patch assay developed. And I
Okay.
don't know all of the results, but so far it sounds fairly promising. Is that right, Søren?
Yeah.
Have you ever tried phagolysosomes? Because they're usually considerably bigger.
Oh,
Are those the swelled ones?
They're
No,
they're
well,
from
naturally.
Ah, right. Okay. Though.
Right.
What did you call them again?
phagolysosomes from a macrophage or a microglia,
Ah,
because you can feed them a bead, and the bead can be 5 microns in size.
oh,
Lysosome fused with it, you then have your lysosomal channels, but on a organelle which is 5 microns in size.
oh, wow. Yeah.
No, I don't think anyone's doing that, but that's a good idea.
We're trying it with
And it has a
Iwan on the
PhD
porta
student.
patch.
Ah, right.
Oh, wow.
Okay.
Cool.
We'll see if it pans out.
Yeah.
But yeah, probably not for screening purposes.
Probably not for
No,
screening.
I also agree, not for screening purposes. I think there you've really got the right idea to have it, the plasma membrane, having it expressed at the plasma membrane, because obviously that's going to give you the biggest or the best success rate, I guess, for high throughput screening. And then, combining it with these other techniques like the SURFR for checking whether your lead compounds actually do what you think they're doing is also, I think, an important step.
Definitely, yes.
I would just like to say again then, congratulations. I hope this has been like a nice early Christmas present for you.
Thank you very much. Yeah.
Congratulations.
Amazing. Yeah, thank you very much.
I'm really
We
looking
are
forward to
actually
hearing
joking
them.
about this, so that it would be lovely to have this as a Christmas present.
Okay, well, thank you very much for your time today, you guys. And
Great. Thanks for the great
congratulations
news.
again and happy
Yeah.
Christmas.
Brilliant.
Yeah.
Thank you very much too.
Thanks,
Yeah,
all.
thanks a lot.
Bye bye.